As Chris says, I'm going to try to give a sweeping overview of a lot of the fundamental work in the time frame about 5 to 10 years ago that we participated in that led us to the era of genome-wide association studies that you've been hearing a lot about in recent years. And you will hear in more detail at upcoming primers. So let's dive right in. To give the background of why we focus on human genetics, it's primarily not about prediction. It's really about developing a much more complete knowledge base for therapeutics and preventions. And basically what this amounts to is the fact that we're trying to counter decades-long trends in which the pharmaceutical industry has gradually gotten less and less productive because the targets, despite all of the great chemistry that's available and the modeling and so forth, are simply not relevant to disease. So no matter how well you perturb them with a chemical, they're not going to have the effect that you would hope they would have. And that's because we fundamentally lack insight into human disease that genetics can give us. It may not be the only way, but it certainly is a tried and true way in which we can gain insight into the underlying biology of disease. And clearly in the case of neuropsychiatric disorders, we are still basically treating them with the same compounds that were developed more than 50 years ago now. And that's obviously not an acceptable situation. Now, it's also clear that genomic information has been, for a long period of time, vastly oversold in terms of its ability to deliver personalized, individualized medical insights. That reality is starting to come upon us, several decades after it was first promised. But is not the primary goal of most of what we engage in as human geneticists, yet obviously we want to take advantage of those things when they arise. So why does genetic variation matter? So, it's more than simply a hypothesis that genetic variation might lead us to actionable leverage points in the biology of disease. So for example, the first two common genetic variants that were discovered as relevant to type 2 diabetes risk were actually in two genes which were already known to be the targets of anti-diabetic drugs, two of the three most commonly prescribed anti-diabetic categories. So this was obviously a retrospective discovery of variation in genes that were already known to be relevant to diabetes and the effective targets of drugs, but clearly it articulates the point that if we were to find many more genes, we would very likely also be exposing new potential targets and leverage points in that biology. Another example I like to give is from age-related macular degeneration. So we knew next to nothing about the underlying biological origins of the most common cause of late adult blindness in our society up until the first genome-wide association studies began now about 6-7 years ago. And since then, we've recognized a large number of genetic variants clustered in an immune process. The complement pathway are really at the heart of the underlying susceptibility to AMD. This has dramatically changed the way that we think about the development of therapies and the study of this disease, because before these studies were performed, there was, as you can see, a great deal of energy put into research into AMD and an even greater amount of energy put into studying this complement process in the immune system, but there were basically no evidence that anyone had ever put those two things together as potentially linked to each other. And now, instead of studying how drugs might work on mice with burned retinas, we can actually study how drugs might work in mice that actually have an age-related retinal degeneration owing to the same reasons that humans have it, perturbations of this complement factor gene. But we can only do these things if we understand the underlying human genetics. So if we take our cues from the human biology, we can really make much greater progress. We can also, in this case, because the genetic effects are so strong and so common in the population, we can identify individual slices of the population that may be at 100 or 200 times greater risk than other individuals who are highly protected from this disease. And as we discover ways of intervening in advance, early in disease or prophylactically before any disease is present, this kind of genetic information might become useful as well. And one final example that's been oft talked about over the last 5 years. Many in the medical community recognize, obviously, one of the challenging things to do is to deliver warfarin, the most common anticoagulant, safely and effectively, because it has a very, very individualized range in which it's effective for an individual, in which you balance the need to perform anticoagulation with the risk of over-anticoagulating and putting the individual at risk of hemorrhage. And so, since there's wide inter-individual variation in how much dose is right in that narrow range for each individual, this has been an opportunity to explore, again, retrospectively, what the value of genetics might be in helping make personalized decisions about drugs. And in fact, as you can see from this regression graph over here, if you take into account the two largest genetic risk factors that have been, or genetic variants associated with warfarin dosage, as well as an individual's gender and BMI and age, you can actually get a pretty accurate prediction of whether they're going to end up on needing a very high dose of warfarin or needing a very low dose of warfarin. And having that knowledge in advance would, of course, help the individuals at the high end who may start out with a dose that's not effective for them, while not putting individuals at the low end at risk of dangerous hemorrhages. So finding genes would be good because we could do a lot with them. And that's beginning to come to pass over the last 5 years in the genome-wide association study era, but we've got a long way to go. But what I'm going to do today is I'm going to walk backwards in history, actually start a long way backwards, and describe a bit about how we find genes and how our struggles to find genes throughout the 1990s led us to a renewed focus on understanding genetic variation patterns in humans and how that led us up to where we are today. So the starting point, obviously, of any genetics lecture can be, if you want, Mendel. And of course, Mendel was the first to recognize that there were these discrete units of inheritance. We now know these are genes, and we know they're connected to each other on chromosomes. That was obviously quite opaque to Mendel back in the middle part of the 1800s. But clearly, this was the first documentation of what we have built nearly everything in genetics on since, that there are transmissible units of inheritance that result in reliable phenotypic differences among offspring and grand offspring and so forth. Now, the first thing that confounded Mendel is the actual thing that we've taken most advantage of historically in discovering and identifying the genes for human phenotypes, and that is linkage. And so why this confounded Mendel was, of course, that some of the pairs of phenotypes, the color of the shell and the shriveled or smooth nature of the shell, for example, as just a cite example of two of the phenotypes you might have studied, were not passed on independently. And so while each phenotype unto itself, like the color or the shriveledness or whatever, was passed on in a perfect Mendelian ratio, there were some pairs of phenotypes that were not passed on independently, but appeared that if you had parents or a parental strain that could produce both colored or colorless and smooth or wrinkled peas, then in fact what you would sometimes find is that for certain pairs of those phenotypes, the offspring were almost always colored and smooth or colorless and wrinkled. And this was kind of annoying to Mendel because it wasn't always the same. There were a few percent of the offspring that sort of went the other way. So it wasn't independent, it wasn't the same. So this was troubling because we were thinking of these things as sort of grains of sand of inheritance passing through the hourglass of time. So of course the resolution came a long time later, and in fact due to observations by these guys, Sturtevant and Morgan, in the early part of the 1900s, in which the answer is obviously that the units of inheritance, or the genes responsible for those two phenotypes, are actually tightly linked to each other on a single chromosome. So they in fact are not passed on independently, even though they are distinct genetic entities which control the phenotype in that particular cross. So as I said, Sturtevant and Morgan used this concept initially to really build the first linkage maps, and this was of a Drosophila X chromosome. And they didn't, of course, have genes. They didn't have molecular techniques, and we're talking, almost half a century before understanding the structure of DNA and anything that we would possibly think of as modern molecular biology. But what they were able to do is, of course, take multiple phenotypes and track the inheritance of those phenotypes through multiple generations of Drosophila crosses, and realized that they could actually construct the linkage map of these chromosomes based on the proximity and the relationships, sort of the mathematical relationships of this flavor when they work out all the pairs and triples of phenotypes. And so this became, over the next half century, a very rich area of analysis, even long in advance still of being able to perform molecular studies on DNA, in which we understood, began to understand how diseases tracked with other diseases or biological phenotypes or blood group markers or protein markers that could be measured, and built up the mathematical foundations of what we call family-based linkage analysis, in which ultimately, decades later, and this really flourished in the late 1980s and throughout the 1990s, we were able to study, in the modern instance, DNA polymorphisms scattered throughout the genome and immediately track down which arms of which chromosomes were essentially co-inherited with the disease, and so thereby localize or begin the process of localizing the identity of the disease gene in terms of its genomic position. And so this general strategy has been the primary driver of a first revolution in human genetics throughout the 1990s, in which thousands of genes underlying countless Mendelian disorders have been identified, including very rare and early onset forms of common diseases such as Alzheimer's and breast cancer and so forth. And this really works quite well, this sort of tracking of co-inheritance of DNA and disease, when, and really only when, there is a direct and deterministic relationship between the genotype and the disease state. So if you only get the disease when you have a certain DNA mutation, and you always get the disease when you have that mutation, then there's going to be this perfect relationship between the inheritance of a stretch of DNA and who gets the disease in the family. This makes it pretty easy if you study 5, 10, 15 families that have the disease because of a mutation in the same gene, to really narrow in on the location of that gene. And that's really what led to this first revolution here, articulated by the pink boxes, that demonstrated for us that we could actually very effectively even get down to the individual gene responsible for a whole host of human disease phenotypes. And throughout the 1990s, as I said, probably on the order of about 1,800 such mapping discoveries were made. And at the same time, despite a much greater effort in terms of money and samples studied and so forth, we tried to do the same, apply the same strategy to families that segregated more common diseases, such as type 1 or type 2 diabetes, or any metabolic, inflammatory, neuropsychiatric disease. And the scoreboard for that effort is represented down here by this sad set of blue triangles and this very different blue axis over here. And so we quickly came to realize, well, it wasn't that quick, but we came to realize that there were some things, those disease entities that had that deterministic relationship between genotype and phenotype, that were very amenable to this approach. And there were many, many other things that were not. And the reason they were not, there's countless reasons why they were not, but certainly if a disease or a phenotype involved the action, whether separate or coordinated, of 10 or 20 genes, and whether those genes didn't directly result in they didn't magically give you an MI at age 60, they altered a lot of physiological features in the body, a lot of biomarkers and so forth, that would lead to putting an individual at greater risk for that MI at age 60. And if there were also many environmental factors, obviously with none of those confounders would our traditional strategy be effective, and lo and behold, it wasn't. So what became clear as we turned the page, past the year 2000, was that we really needed to have a more systematic approach to study the role of human genetic variation in disease. And this is at a time when we were still in the process of finishing the first draft of the first human genome, and so we really didn't have any of the tools and capabilities that we think of today. And in fact, at that time, we couldn't even remotely imagine that in 10 years we would have the ability to survey the genomes of thousands of individuals, that we would have the ability to sequence the genome of an individual in a matter of days. But it was clear that in order to make a stab at dealing with some of the complexity of the phenotypes that we were looking at, even though they were extremely heritable, it was clear that we didn't have these resources. So basically, an understanding of genetic variation patterns in the human genome, laboratory tools to test it comprehensively, to really be able to test the polymorphism content of an individual in more than 10 sites, but maybe 1 million sites. And then a new suite of analytical methods that can go beyond some of the things that we'd come to know and love with the family-based linkage approaches. So we began, as part of the Human Genome Project, and then as sort of spin-off projects, to assess the DNA variation content of the human genome. So this actually, in the year 2000, was a completely unknown question. How much variation is there in the human genome? And so this is where we started our investigation. And we began to ask this question because as part of the Human Genome Project, of course, the genome was cut up into many different pieces, and those pieces were then sequenced and overlapped back together. But of course, the Human Genome Project wasn't one person's sequence. It was a library of sequences from many, many different individuals. And so when those two pieces overlapped, we got an eye to, when you take two chromosomes and line them up, how much variation is there? And when we began to ask those questions, when we compare any two chromosomes, about once every 1,000 base pairs, we saw a difference. Most of them were just simple single nucleotide changes, as I'll show you. But what was interesting to us was that when we would find a difference and then look at a larger sample of individuals, say just all of us in this room here, 9 times out of 10 or 19 times out of 20, that difference that we saw in the two chromosomes was actually a common DNA polymorphism in the population, that we'd find multiple copies of each of those two alleles, suggesting this was a very old DNA polymorphism, not a private new mutation to one or the other of those individuals or their recent families. So most of the DNA variants that we would find would be these very uninteresting and benign-looking single nucleotide changes as we walk through the genome. And so this already began to articulate for us that we would have one challenge, is that even if we could begin to catalog all this variation, it really didn't come with any markers to tell us which ones were going to be interesting, and we can start to think that there are going to be millions and millions of these across the genome. It's going to be very challenging to actually get to those next steps of associating them to disease. So genetic variation, some in bits and pieces in some places was a little bit more interesting than others, because while I said most of the DNA variants were these sort of uninteresting and benign single nucleotide polymorphism, there were many other types of DNA variants that we began to encounter and catalog, and some of them, such as large deletions and duplications and inversions and things, we never expected to find as common polymorphisms, but in fact, often even in the cases of some very large deletions and duplications, these are actually, simply common and largely benign polymorphisms in the population. So this seemed like a lot of variation when we began to think of, boy, it takes us a lot of effort at this point in time to, sequence one gene or to DNA type, 20 polymorphic markers in a forensics lab, to think about the vast millions of DNA variants across the genome. But in another sense, we also began to discover the fact that humans are actually among the least diverse organisms that walk the planet, with the possible exception of a few species that are in very restricted contraction owing to almost being extinct. Humans have about the least amount of DNA diversity of any organism that we'd study. And so while a difference every 1,000 bases when you compare two chromosomes sounds like a lot, it actually isn't compared to almost anything else that counts itself as part of the same species. But it is a lot in another sense, in which there are, on the order of at least 10 million, and now the 1,000 Genomes Project is really cataloged on the order of about 30 million sites of common DNA variation. The only difference is whether you define common as down to 5% or down to 1% or something. They're just words. They don't have any special meaning. But there are a lot of DNA polymorphisms that are at a frequency, whether it's 1% or 5%, that tells you they're not under severe adverse selection from being deleterious and not being permitted to exist in a lot of people. We, at the time, began to look across populations and quickly saw that, as everyone had figured and as anthropologists had told us for a long time, there was strong evidence in the DNA content of the human genome when we compared populations for the out of Africa hypothesis, in which nearly every DNA variant that we found in any population was found in Africa, and there was also a lot of additional DNA variation that was found only in African populations and not found in East Asia or European populations. And that's simply a reflection of the fact that not everybody came out of Africa, obviously. Only a very small subset of the population in Africa came out of Africa. So this, was a large number, but tractable in terms of, how much DNA variation there was. Tractable in the sense that we could actually hope to catalog and understand it someday. The other compelling observation that we made in the early part of these studies was that the DNA variants that we saw and discovered in any number of ways, but let's just stick with the comparing two chromosomes and finding a DNA variant, and generally it's old. Generally it's old, but generally it's also unique, and so there's a unique signature that we can see when we encounter a new DNA mutation. And that signature is we sample individuals in today's population, and we might find two individuals or two chromosomes here that share a very rare DNA mutation. And so what you can imagine happening is that if this shared DNA mutation is a unique historical event in the human population, then what this means is that at some point in history there was an individual. That individual got a new mutation when they were born, or prenatally or whatever. This new mutation they then passed on to their offspring and their offspring and so on and so on. And that leaves a signature, not simply that this base happens to be shared by those individuals, but the reason that these two individuals on these chromosomes got that particular DNA variant was because, of course, they have a stretch of DNA that's identical, that dates back to this common ancestor in human history. I mean, every piece of DNA has a unique and great ancient history, and so for this small region of the genome, these two people are actually related to each other. And what that means is, of course, that when you compare their chromosomes, they may be different at one base in every 1,000 for the majority of their the stretch, but for this region around this shared mutation, they are identical. And so if that shared segment is only 10 or 20 generations ago, it might still be quite large, maybe 1 million bases long, in which there's absolutely no DNA variation. And so that's a signature that we can tap into to find, and we had tapped into this signature in studying severe Mendelian diseases in isolated populations, and we could take advantage of the fact quite distinctly, and this is just sort of done up in a little bit more detail, but let's skip that for the moment. But we had taken advantage of this in the study of rare diseases in isolated populations by saying, hey, if we're in an isolated population and we take the 10 people with some extremely rare and severe genetic disease, then it's likely that they all have the same mutation in the same gene inherited from some founder in that population. And in fact, we could find the genes effectively simply by screening their genomes and identifying where in their genomes are all of those people carrying the mutation identical. And this is in fact a tool that was used repeatedly throughout the 1990s in the end game. So after that family-based linkage analysis, we'd get down to a region of the genome, and we still obviously didn't have the technology at this time to sequence genomes or even small stretches of genomes. But what we did have the ability to do was to study DNA polymorphic markers, say every couple of megabases throughout the genome. And in the case of CF and in the case of many, many other disorders, the identification that most of the individual chromosomes from CF patients were identical to each other for a significant stretch of the chromosome 7 region in which the family-based linkage had focused everyone's attention was instrumental in rapidly narrowing down to the identification of CFTR as the causal gene. And this has been used, as I said, countless times. But again, it was thought to be a feature of the study of highly penetrant rare mutations in isolated populations, because those mutations would be recent and it would be easy to track back to a common ancestor that produced the individuals who carry the disease. It was thought to have essentially no relevance to the study of common disease because, of course, common disease has many, many different factors involved in it, and no one would think there's a recent common ancestor of everyone with type 2 diabetes. That would be silly. So when we began to explore genetic studies of inflammatory bowel disease around this same time, the early, right around 2000, 2001, it was with a mindset that linkage analysis could give us a clue, but probably wouldn't. But maybe we'd give it a shot, and with a mindset that the idea that we might find a common founder mutation was probably pretty silly with respect to a common complex disease. So the inflammatory bowel diseases, Crohn's disease and ulcerative colitis, are chronic inflammatory diseases of the GI system. They have a significant genetic component, as by the significantly increased risk to individuals if they have a sibling with Crohn's or colitis, and very high concordance rates among monozygotic twins. But at the same time, it was clear that these were not single gene traits, because there had been some linkage studies done throughout the 1990s that put this in the category of the complex things that linkage wasn't really working for, as opposed to the ones in which it was. So we performed one of these linkage studies in the late 1990s and began to focus in on a region on chromosome 5 because in our earliest onset Crohn's disease patients there was a strong signal. And this was a trick, if you will, we had learned from the identification of the BRCA1 and BRCA2 genes, in that a common disease is not likely to be amenable to these linkage approaches as I've been describing to you. And such was the case for breast cancer. But when researchers focused in on families with particularly early onset breast cancer, this was a scenario in which there were higher penetrance mutations, and linkage studies were actually successful in that subset of the population. So we tried this and got a pretty reasonable result in this region of chromosome 5, so we decided to dive in further. First, we looked at individual polymorphisms in the region, got some hint that some polymorphisms in this region containing a number of interesting cytokine genes might be associated to Crohn's disease, and so began what was at that time a very laborious process of discovering the DNA variants in this region and then testing them in hundreds of patients and controls. Because we didn't have a genome reference at this time for this region, so we actually sort of had to, begin to construct one, or help fill one in at the same time as we were even discovering DNA polymorphisms in the region. So we began to perform discovery of single nucleotide polymorphisms and indels and other things across about a 500 kilobase region containing those interesting genes, and then we pulled 103 common polymorphisms as just representative of polymorphisms we had discovered from that data set, and began to look at a few hundred individuals just to understand what the variation patterns looked like across this region. And they were, curiously, a little bit more interesting than we had figured. So what we first began to do was to take windows of polymorphisms and walk across this region. So we took windows of 5 or 10 DNA markers and slid them along to see how much diversity there was in terms of how many different combinations of alleles or haplotypes or sequences, if you will, were we seeing across different parts of this region. And there were some interesting dips in these patterns that told us there were some regions that had curiously low diversity in them. And we didn't really know what that meant because we thought, there was going to be a variant every 1,000 base pairs and so forth, and that there would be a lot of different combinations of those variants. And so what we saw was very curious. So here's a region, what part of that subregion, that first one that I just pointed with the arrow, where we found 8 different single nucleotide polymorphisms, but they somehow all seemed to be highly linked to each other in a curious fashion, in that there were only 2 different sequences. So instead of 2 to the 8th combinations, there were only 2 combinations of those polymorphic alleles at each of those bases that were common in the population. And this was just a random control population from the metropolitan Toronto area. It had no special isolated population features. And it stretched over almost 100 kilobases. And so in theory, there should be no such observation because of the number of generations back to common ancestors in the human population and the amount of recombination that we knew happened in the human genome. And we had a pretty good feel for how much recombination happens in the human genome because the one thing you can observe is how much recombination is there in, we had accurate linkage maps for the previous decade telling us just how much recombination happens in each generation, and so we can obviously extrapolate back to tens, hundreds, and thousands of generations. We also began to see that the variation patterns we were seeing occurred in segments, in that there were these long stretches where there were only 2 combinations, but then seemingly some sort of a breakpoint in which we would see all combinations, again, not randomly assorting the individual polymorphisms, but sort of 2 of these segments stuck together in which you could see not only a continuation of the same patterns here, but sort of a crossing over. And this, of course, was quite recognizable as, in fact, crossing over itself. So the first haplotype maps, or HapMaps, that we constructed were of this region of chromosome 5q, in which we could actually dissect out stretches where there was very limited sequence diversity in individuals, and then these crossing over points where apparently meiotic recombination had acted to assort what were sort of ancient sequence patterns that had been just mutating but not recombining for a long period of time. We did in fact discover that one of these patterns was reliably associated to Crohn's disease, and that's something that's since been replicated a number of times, and that's not really the point of the discussion here. But what was the point, and what was the more lasting impact of that project for us, was that it began to articulate that not only could we catalog the DNA variants in the human genome, but that there might be some great utility in cataloging their relative patterns and relationships, these haplotypes. Because if you think about it, one of the promises that this information and this representation of the information holds is that it tells us that here's a 100 kb, or nearly 100 kilobase stretch of the human genome in which there's hundreds and hundreds of DNA variants that you might consider relevant to disease. And there's a couple of interesting genes contained in that region. But there's really only 4 different sequence patterns that are commonly found in this population. And so we could essentially imagine that just simply by typing 2 or 3 DNA polymorphisms in this region, we could actually reconstruct which red, blue, green, or purple chromosome are carried by each individual and perform a comprehensive study of the association of this region to disease simply by looking at those 2 or 3 polymorphisms, not needing to sequence the whole 100 kb and tally all thousands of the variants that are in there. So this was the idea that led us to the concepts which have, that genome-wide association studies were built on. And those concepts being that, a catalog of DNA variants that shows us those patterns might be useful for the interpretation of genetic association, might provide a great shortcut in which, instead of sequencing whole genomes, we could actually look at a very small number of polymorphic bases, and that this information might be useful for designing those experiments in a very efficient and accurate manner. And so the HapMap project was essentially a spin-off of the Human Genome Project, if you will. It sort of began right at the tail end of the Human Genome Project. And if you think of the Human Genome Project as giving us the fundamental baseline of information with which we perform many experiments today, the layout of all the genes and so forth, the one thing it didn't give us was, of course, the one thing that medical geneticists are most focused on, which is where are there differences in individuals' genome that lead to phenotypic consequence. And so this was building the layer of inter-individual variability on top of the human genome reference. And so I'll dive into the description of this study in a moment, but the focus of the design of the study and throughout its execution was reflected, of course, in just the way I've described it to you today. It's meant to be a tool to enable and empower the study of complex human disease phenotypes. And this is the way in which we carried out the project, and this is the way it's been used since its completion in 2007. And so essentially this was the community's response after a number of years of struggling to and not finding genetic factors for complex disease, and for beginning to get some understanding of human genetic variation and what it might take to get us to discovering genes for complex disease. So the data from this project was, of course, always something which was made available to the community in real time, and we took our cues from the Genome Project, and many projects have obviously followed in the footsteps of the HapMap project with respect to providing basically real-time, full community and public access to the data as it was being constructed, so that it could be used and integrated into labs throughout the world. We also developed a number of analytic tools that are still in wide use that as we often struggle with these large public research projects, they generate enormous amounts of data that are stored on some FTP server somewhere, not always in the most useful and accessible format to the clinical researchers and the biological researchers who are the intended user base of this information. So we developed tools to make the data more accessible, make it more integrable with the way in which geneticists were conducting genetic research. And the data began to tell us interesting stories. Clearly, we began very quickly in performing this project to get some understanding of why it was we saw those patterns in chromosome 5q31. And some clever methods were developed at Oxford for taking the population data and inferring a recombination map. So rather than having to just study individual single 2 or 3 generation families and understanding the recombination patterns in those families, being able to infer it from these linkage disequilibrium patterns. And it was clear that the reason we saw all this structure in DNA variation in these haplotypes was, of course, because meiotic recombination was not occurring homogeneously through the genome, but was occurring primarily at intense and punctate hotspots. And so the second part of the presentation today focuses on understanding what the HapMap is and why it's useful and what we use it for, what we continue to use it for. I mean, this was a project that was done from like 2002 through 2007, but it's provided a tool base and a set of concepts that we still build off of today. So the HapMap was, of course, as I said, meant to survey, not in a comprehensive fashion, but in an initial fashion. We were at a time when we really didn't understand human genetic variation at all. So the little that we did understand suggested to us that if you had to pick 3 places in the world to sample human diversity, you would definitely want an African population, and you know, for lack of anything better to do, you'd probably want to take 1 European population and 1 East Asian population to capture some of the interesting major, you know, migrations in human history. So the project was launched in 2001. It really didn't get going into full production phase until 2002, 2003. There was an initial phase completed in 2005, and part of the analysis of that initial phase was to determine just how far we had to go with this activity to make it fully useful, and I'll describe what that means in just a moment. So one of the questions that we asked early on in the project was, well, it sounds good to survey DNA variation and make a map of it, but we couldn't do this by sequencing genomes. We just finished sequencing 1 genome at the cost of about 10 years and several $billion, so that clearly wasn't going to happen. We needed to identify polymorphisms and then test them, but how many before we got a map that was relatively complete? And so there's a couple of different ways of looking at this. Obviously, you know, one of the things we wanted to do was to have a map that was relatively complete down to fairly low frequencies, say down to 5% frequencies, so that we could at least say we had a fairly comprehensive view of patterns of common DNA variation. And so one could sequence small regions of the genome and then say, all right, what have we just found and how much of it is already known polymorphisms in dbSNP and so forth. And you see when we sequenced some of the individuals from the Yoruba population in Nigeria, that almost all of the common DNA variants that we found in the Yoruba population were already recorded as polymorphisms in dbSNP, or turned out to be in high linkage disequilibrium, or highly correlated to variants that were in dbSNP. So this would tell us that for these allele frequencies up here, 20% and above, we could do a pretty good job simply by picking the polymorphisms that were in DB databases, public databases, and then testing them in the population that we had ascertained from Nigeria. For lower frequency polymorphisms, it was clear that quite a number of variants that actually existed in this population sample were not already known and were not at all, you know, tested or proxied by anything that we could ascertain out of dbSNP. So more sequencing was required, but it wasn't a huge amount to get us to a point where we could start to build up haplotype patterns across regions and so forth, and expect that they largely represented most of the common DNA variation. Of course, there's always going to be sprinkled on common sequence patterns rare variants, and that's not what this project was about. Technologically, we didn't have the ability to assess that. This is more the focus of the 1,000 Genomes project, which is essentially the direct descendant of the HapMap project, but using sequencing technology to make a more complete assessment of variation. So as I said, we recognized the recombination hotspot patterns and how we could use those, but just making pictures and maps wasn't exactly the focus of the activity, because as I said, we wanted to have an activity that was delivering a tool and delivering strategies that could be used in association studies. And so we wanted to take the view that we set goals and targets for completeness in this activity that were relevant to what we would want to do in the future with respect to association studies. And so we sort of anthropomorphized ourselves and said, well, the real question about how good a map is if there is a disease-causing DNA variant in a particular region where our map is meant to cover, does our map actually include that variant, or does it include a variant that's tightly linked to that variant such that the association test would give you a positive result? So this is how we began to assess the coverage of the HapMap, and I think you can tell the assessment of the coverage of the HapMap is better than my ability to line up text on a slide. But what we were focused on was making sure that the majority, in this case almost 95% in the non-African populations, just over 80% in the African population, of DNA variants were assessed highly accurately, and that the average DNA variant had a very strong, highly correlated proxy on it. And so these are just the standard correlation coefficient telling us that if we pick a random common DNA variant anywhere in the genome, the average correlation to a SNP on our HapMap was 0.97. So this means that if you performed an association study just with the SNPs on the HapMap, you would actually do a very creditable job of discovering genetic associations without much loss of power, compared to a strategy that involves sequencing and ascertaining every single DNA variant. And that was really the goal. So the project itself was focused on a number of different applications. Some of these have come to be, the far and away the wide and continued uses of the studies. At the time, some of these earlier applications were actually much more important to the field. So the idea of tagging is not something that we have to worry about or talk about much anymore, but it took up an enormous amount of focus at this time, because this is what we primarily needed to do when we were performing an association study. Yes?
Quick question. When you say that the SNPs are similar between different populations, but the sizes of the haplotype blocks could be very different. Because they're a combination of spots in different populations. So how do you account for that when you just make a haplotype map for all ethnicities?
Good question. So we'll come to the assessment of the HapMap with respect to that question shortly, because that is a very important one. With respect to the general difference between populations within Africa and populations out of Africa, you can see both the impact of the fact that there's a lot more variation in the population in Africa, and the fact that the recombination has been essentially going on for longer, such that the LD patterns are smaller, they don't extend quite as far. Both of those are reflected in the fact that a fixed map would always describe the variation out of Africa more completely than in Africa, simply because we would need a larger and denser map to get to the same point for an African population. But we'll come back to the other populations, not these three that are in the study, because it's a very important question.
You still make like one map for all.
So we made three different with three different maps for each of these populations and a unified map. The variants are, as I said, by and large, considerably shared. So there's very little unique variation to each of these populations. And so the polymorphism catalog and map there's one, basically for all variants, whether they're polymorphic in one, two, or all three of these populations. The linkage disequilibrium pattern maps we created separately for each of the populations. Because of their very different history, the recombination patterns can be a little bit different. So tagging is the idea of, if we have this complete set of DNA variants, can we choose just a few of them to accelerate our genetic association studies or make it a lot more affordable, and also non-redundant, basically to save money and to make efficient what was an extremely inefficient but also uncertain process. When we would pick some polymorphisms in a gene to see if it was associated, you would be testing those polymorphisms, but you'd have no idea if there were other polymorphisms that you could have found that might have been associated, but you didn't test them. And so the idea here was trying to get us to a more regularized and rigorous place, and this eventually we don't have to do anymore, because it was taken to the genome scale and integrally used, and still is, in the design of genome-wide association laboratory tools. So this is colorful, but it's largely eye candy. The idea of tagging is a relatively simple one, and it's in its simplest instantiation, it amounts to this. What I told you about, hundreds of variants and having only 3 or 4 recombination, 3 or 4 different sequence patterns, that's true, when and only when the scenario is as follows. There are basically a large number of pairs and triples and sets of 10 or 20 polymorphisms that are genetically identical to each other. So on every chromosome where these are just representations of 4 different patterns, 4 different haplotypes or chromosomes, if you will, every chromosome that has an A at this particular position has a G at this position. And every one that has a T at that first position has an A at that position. So they give you the identical information in a genetic association study or any other type of genetic study. So if we were trying to be efficient about what we genotyped in a study, we clearly would not need to genotype those polymorphisms that I've just blanked out there, because of course they are redundant with other ones that we are genotyping. So that's one way in which we can very rapidly and without much cleverness save on our genotyping bills. We can go further, because beyond the pairwise tagging efficiency, which in the first version of the HapMap project we were able to use to estimate, if we wanted to do that across the whole genome, how many polymorphisms would we need? And these numbers, if you want to have, just eliminate fully redundant markers, or you're allowing yourself to eliminate more markers by relaxing that correlation coefficient, you could actually, for only a few 100,000 DNA polymorphisms, do a reasonable job of a genome-wide association study. So what was also clear, though, was that the predictive value between one polymorphism and another wasn't contained to the most simple, pairwise identity relationships, that there were, in fact, highly reliable and predictive relationships between pairs of polymorphisms and other polymorphisms. So for example, this polymorphism over here, in which one haplotype pattern carries an A and the others all carry a C, is entirely predicted from the haplotype pattern of the first 2 polymorphisms. So if a chromosome carries an A here and a G here, it carries an A here. If it carries any other combination of alleles at those first 2 sites, it carries a C here. And so, in fact, if we were doing that, trying to save money, we don't even have to test polymorphism 6. All we need to do is test the first 2, and then remember that when we come to our association study, we need to remember that we also remember that the AG haplotype here actually represents another real DNA variant that we wanted to test. And so when we come back to our test of association, we would remember that fact and include not just the first SNP and the third SNP, but this combination test in our test of association. And so this is still essentially the process that's going on under the hood in terms of genetic association studies that we conduct today, and I'll come to that in a short bit with respect to what we call imputation strategies, and you'll hear more about those in upcoming weeks. So Paul de Backer, when he was working with David Altshuler and myself on this project a number of years ago, took it upon himself to see how accurate and efficient could we make a genome-wide association study in theory. This was still all theoretical because we certainly didn't have the ability to test hundreds of thousands of polymorphisms. But what was particularly exciting was that Paul was able to demonstrate, using the entire HapMap and how well it covered DNA variation in general, that if we chose very, very intelligently, based on sort of the principles in those last few slides, the set of DNA polymorphisms from the HapMap, we could construct a pretty reasonably well-powered genome-wide association study with 300,000 well-chosen tag SNPs, as if we had been testing the entire polymorphism content of the HapMap. And so this was, the time at which the companies, particularly Affymetrix and Illumina, began to, had simultaneously really accelerated the technological development of arrays that would allow us to potentially study hundreds of thousands of DNA variants. And in fact, the first Illumina genome-wide association product contained 317,000 SNPs chosen with the methods that Paul had described, and you can see that number that they ended up with for their first major genome-wide product is quite resembling this figure that Paul came up with in this paper. So we don't actually pick tag SNPs anymore, so I'm not going to bore you with telling you how we used to pick tag SNPs because, as I said, it's no longer really an issue for us because once companies who produce genotyping products are able to tap into all this data and these methodologies, they're essentially doing this process for us. And so starting around 2005, 2006, genome-wide association arrays became, began to become affordable and distributed to the research population, and we no longer had to worry consistently about picking tag SNPs from the HapMap because that was done sort of once and for all. So to come back to the question you asked a few minutes ago, a critical, one final critical piece of analyzing whether or not the HapMap would be useful was cleverly pointed out a moment ago, which is we built a nice map of some European people who lived in Utah, some Yoruba who lived in Ibadan, Nigeria, and some, well, all right, they were broadly chosen Chinese and Japanese individuals from major metropolitan areas. But how well would such a map constructed in those individuals represent the broader population? And so another important benchmark in determining the completeness of the HapMap was some studies that we did in a number of other populations, but I'll just highlight a few of them here. Taking this map of DNA variation built in a bunch of Utah residents with European ancestry, we asked, how well does that map explain variation in a different group of Utah residents with European ancestry? And how well does it explain variation in self-described whites from Los Angeles, quite likely a more diverse group than the Utah group, and a Swedish-speaking isolate on the west coast of Finland, quite possibly a less diverse group than the Utah ancestry, but a different group. And in point of fact, when the HapMap identified that two polymorphisms were completely or very highly correlated in this original sample, they turned out to be completely or very highly correlated in these other samples, and really with very, very little degradation of the predictive value. And so with respect to the recombination patterns, the historical recombination patterns that are represented by linkage disequilibrium patterns and haplotype patterns, while they're clearly not completely shared across major ancient ethnic groups and racial groups, they are within populations of recent European descent, recent East Asian descent, and recent African descent, quite comparable and similar, such that the map we were constructing was complete not only on the variation level, but also on the recombination level. So, pressing question, as I said, I've sort of already talked through this, so I'll move through it quickly, was, as we were completing the map, was, is it actually possible to perform a whole genome association studies? And so, of course, the answer to this is, of course, it is, because we've been seeing them now for a good 5+ years. And we partnered, the project partnered, and we partnered with Affymetrix, Illumina, and anyone else who was willing to, design such things in a way that didn't, provided no, financial value to us, but just because we wanted these companies to be developing tools that were maximally useful to the research community, to make sure that they had access to and understood all the data that was coming out of the HapMap project and how they might use it effectively to design these products. And of course, the HapMap has been, of course, a very instrumental tool, from this time forward for the design of genotyping arrays of first 100,000, then 300,000 and 500,000, then 1 million and 2 million SNPs. We could help, the HapMap was particularly helpful in assessing the coverage and completeness of any of those polymorphism sets, because if they chose some markers from the HapMap, there are of course many, many other markers on the HapMap that they didn't choose and could serve as an assessment for how well their product covered the genome. And so that's exactly the kind of analysis we began to do with each time a company would put out a new product, we would say, how well does this product cover the genome? And so we'd perform almost exactly the same types of analyses that we described for selecting the markers, now retrospectively. So we say, all right, we're not going to pick markers this way. Affymetrix has picked these two markers from this region of the genome for us. So what can the HapMap tell us about how much information those markers cover and what we should do with it? And of course, when you then look at the entire HapMap, you can then fill back in the information, even in the predictive fashion that I described a minute ago, in terms of using the HapMap to identify combinations of alleles that predict other alleles that are in the reference panel, but not genotyped directly on the arrays. So we were evaluating these products as they came out. And as we discussed with respect to the map, obviously is the same thing. Any fixed genotyping product was going to do a more complete job of assessing variation in a European population than in an African population. And happily, there have been now renewed efforts in recent years to develop arrays with specific African focus content to really make up these gaps, such that we can do genetic association studies now effectively and almost equally in all populations of the world. So, through that. And the point about using the information in the HapMap to construct these more predictive multi-marker tests, we demonstrated early on in very crude analysis that we could dramatically increase the coverage from any fixed set of markers provided by Affymetrix or Illumina by combining those markers intelligently and adding those tests of association, so moving us from maybe 60% of the genome being covered to up to about 80% of the genome being covered. And these approaches have gotten much more computationally sophisticated over the intervening years and have become what we now refer to as imputation techniques, where any set of markers that we happen to assess in a genome-wide association study, we can look at reference panels provided by the 1000 Genomes Project or the HapMap and extrapolate out the genotypes at all the other sites that we didn't directly genotype in a fairly accurate fashion, and then utilize those extrapolated or those imputed genotypes in our association studies. This is now again, standard operating procedure. We don't have to think about it much. It's well instantiated in pipelines and software tools. But this is the underlying origin of a lot of what we take for granted when we see genome-wide association papers, or even when we conduct genome-wide association studies ourselves. So just in the last section of the talk, I'm going to walk through some of the ways in which we utilized the HapMap at the time, and some of these are still actually germane to the ways in which we think about and approach genetic association studies in general. Obviously, linkage disequilibrium patterns, and now much more so with the 1000 Genomes Project, allow us to identify when we see a positive association in an association study. We figure now, it doesn't necessarily pertain to that particular DNA variant, but we need to, as a routine part of our thinking, go through these reference databases and say, I believe this association, how many other polymorphisms are tightly correlated with this one, and create that set of potentially causal variants for functional follow-up studies. And that's a concept that is now sort of deeply ingrained in the field, at least it's still not completely as ingrained as it should be if you read some of the recent ENCODE papers, but basically genetic association results we have to assume don't pertain necessarily, we don't know which SNP they pertain to in these domains where there are many, many SNPs that are highly correlated to each other. So one of the first genetic genome-wide association studies came out while we were finishing up the HapMap, and it actually proved the point that these reference databases were going to be useful for this feature as well, because while the original polymorphism was on, in the Affymetrix, an early version of the Affymetrix genome-wide association arrays, only 100,000 polymorphisms, it was clear that when we were to take that polymorphism and then look in the HapMap at the much, much denser array of millions of markers that we had compiled the patterns for, we could then articulate a very clear list of polymorphisms that were highly linked to that individual SNP that happened to be on the Affymetrix array, and this was used to then very quickly target what the ultimate causal variant was within this set here, which was a very nice and easy to study coding variant in the CFH gene. What has been even a more powerful strategy is the idea, and this is something we still do today, that these patterns of variation that we cataloged in the HapMap and that we continue to catalog in the 1000 Genomes Project are integral tools by which we can sew together the information from different genetic association studies. So one of the challenges of the day when we were constructing the HapMap was that when individual groups would perform genetic association studies on single genes, they would take 5 polymorphisms or 10 polymorphisms from dbSNP and genotype them in their samples. And then, somebody else would come along and say, that seemed like a good idea, that gene sounds interesting, we'll do the same study, but they'd end up picking 5 or 10 different polymorphisms. So then, when you sit down to read these papers, you say, oh, here's RS whatever, and then this next group says, oh, we studied this gene too, and we found RS whatever. And then you're sort of like, well, those are different polymorphisms, so how do I know if there's a consistent story to be told here? And so the HapMap really provided the glue by which we can view genetic association studies across different populations, across different research groups, and across different technologies. Because the magnification of that problem came shortly thereafter when some people started performing genome-wide association studies on Affymetrix arrays, and some people used Illumina arrays. They had completely different sets of markers on them. And so all of a sudden, what was sort of a simple problem was, well, if you're studying this gene and I'm studying this gene, maybe we can get together and agree on what polymorphisms to test. Couldn't be done manually in any sense. And so the HapMap provided an automated way by which we could ask questions like, well, if you tested this polymorphism over here and I tested this polymorphism over here and we both saw that it was associated to phenotype, we could immediately see that, that's the same association, those are consistent, we can now go and celebrate a discovery. However, if you had studied this one and said it was associated and I was studying this one and said it was associated, we say, that's not really consistent at all, and we probably haven't necessarily discovered anything at this point. So one compelling example of how we used this was a very popular set of studies came out in 2002, 2003, 2004, 2005, surrounding the potential association of a gene called dysbindin, or DTNBP1, and schizophrenia. There were a large number of studies, and you can go through the literature. There's a very large, literary history, and I use that term accurately, literary history, on the relationship between this gene and schizophrenia. And what was particularly challenging at the time was that we had no way of lining up any of those studies because they all used a handful of markers that were different. Maybe they overlapped occasionally, but did not provide any consistent, easy way of reading the literature 5, 10 papers in to determine whether or not there was a consistent pattern of association. If 5 or 10 papers all saw a weak association and it was consistent, then they may have actually discovered something genuine. However, if they were telling a different story from each other, we couldn't really tell that at the time. So using the HapMap, we built a simple, linkage disequilibrium patterns were quite extensive across this gene. We built a simple phylogeny, and this is just sort of a mutational phylogeny of the DTNBP1 gene, where we could see what the ancient haplotype pattern across the region is, and we could see, when each of these, the current day haplotypes that we observed in the population, there were about 5 of them, were spelled out, and we could see directly how mutation had arisen in an orderly fashion to create those patterns. So we said, all right, this is very simple. If all of these, association studies were saying the same thing, this should help us sort it out. So we began to then go through the literature on this dysbindin gene and found that the original authors had published 2 papers that described that these DNA polymorphisms that tagged this particular haplotype pattern were associated with risk of schizophrenia. So it's a simple hypothesis. A next study was done, this was done in Irish pedigrees. Next study was done by a German group, and they described that they had replicated the association to dysbindin, but they described risk as being conferred by these haplotype patterns over here, which was in fact not consistent, but it was described as a replication study. Then several other groups performed replication studies, also defining the replication as positive, but declaring risk to this set of polymorphisms over here. Other groups published other replications of a different nature. And ultimately, there was a group that published a study that said there was replication, but the risk was to everything else but the original haplotype, which meant they had actually defined that the original haplotype was protective rather than conferring risk, yet still considered that this was considered at the time to be somehow consistent evidence of an association pattern, which it in fact, of course, is not. When you put all these studies together, there's absolutely no association between dysbindin and schizophrenia. And we sort of use this as a vehicle to explain how one should really think about genetic association studies in a more rigorous fashion, and how you could utilize the tools that were being developed by the HapMap project and so forth to help sort out some of what was a very spotty at best literature with respect to association studies in many phenotypes, but especially neuropsychiatry. And as I said, it's not so much of a problem that people are performing 5 SNP candidate gene association studies anymore, but on the global scale, the activity that we engage in, computationally every day, and it's a pretty computationally heavy one, is utilizing the HapMap and the 1,000 Genomes project to integrate genome-wide association studies done on these different Affymetrix and Illumina and so forth typing platforms, and that each genome-wide association study can now be, sort of inferred onto this common high-density map, allows these 2 studies to be added together. And that process of being able to combine genome-wide association studies has been the primary driver in the last 5 years of discovery in human genetics. And we moved in a couple of different disease areas. Here's type 2 diabetes and Crohn's disease, from studies of candidate genes and genome-wide linkage, which were largely but not entirely unproductive, to very rapidly in 2006 and 2007 individual genome-wide association studies. But honestly, the main progress has been made in these diseases in the time since then in terms of combining multiple genome-wide association studies. These numbers have continued, and we can't, they no longer fit on the screen, because in Crohn's disease and ulcerative colitis combined, there are now 163 genome-wide significant associations that have really begun to lay bare the underlying biology and genetics of these diseases. And so while the challenge that we were faced with in 2000 was that the genes, for Mendelian disorders were coming out fast and furious, but we had made very little progress in complex disease, and even that continued to be the case until 2005. Genome-wide association studies have obviously dramatically reversed that trend and have really revolutionized genetics for complex diseases and common human phenotypes in a way that we couldn't have even hoped would be this successful just a few years ago. And of course, as a reminder, that's of course only the first step in an important discovery process, because where we're moving right now in Crohn's disease, in diabetes, and many other phenotypes, and soon even in schizophrenia, will be to move from the discovery of those DNA variants through a much deeper understanding of their molecular role and their role in disease processes in the body, so that we can ultimately deliver models and an understanding that can be therapeutically intervened. And so with that, I'll just thank the group of individuals, some of whom you know, some of whom you may not, who were integral to the progress that David and I spearheaded in this HapMap consortium 5 to 10 years ago. And look forward to using this as an introduction to genome-wide association studies that you're going to hear a lot more about the technical details of in the next few sessions. Questions? Yes?
This might require a longer answer than you have time for, but I'm just wondering, in general, how many of these principles carry over to indels as opposed to SNPs?
They basically all do.
Mind
Oh, well, repeat the question. Good question. The question was, how many of the principles that I'm describing carry over to insertion-deletion polymorphisms instead of just SNPs? And they basically nearly all do. We use SNPs not because we think they're more likely, they're actually less likely to be functional, but because they're the most convenient experimental proxy. It's clear from the work of the 1,000 Genomes Project that the patterns of linkage disequilibrium, frequency, etc., for insertion-deletion polymorphisms don't look any different than those for single nucleotide polymorphisms. We still, though, as an aside, struggle with the identification, the accurate calling, and the genotyping of those polymorphisms compared to single nucleotide changes. And that especially pertains to the sequencing data, where we're still actively working on methods for improving the calling of those indels. Yes?
Could you speak a moment about rare variants, and in particular, like if you consider like an individual, roughly speaking, like how much of variants are common, how would you define it? How much is rare?
So in an individual, so right now, if you take the information in the 1,000 Genomes Project and you take an individual not in the 1,000 Genomes Project, then much more than 95% of the variation in that individual is contained in these reference databases. You can turn that coin over and look at it a different way. If you were to sequence 1,000 people, then stacking all the variation together, now you've reached a point where probably half of the DNA variants are actually singletons in that set of 1,000 individuals. So while each individual carries only a small, unique or very rare content, when you pile up a large group of individuals, the amount of rare variation is quite profound because everybody's rare and unique variants are different. So those numbers keep adding up, whereas we see the common ones, and there's the same number of common ones no matter how much you build the pile. Yes?
So there's obviously this sort of effort these days to do personal genomes, everybody gets their genome sequenced, right? So, but if the HapMap can essentially tell you, you can just, you can take every SNPs and that's pretty much going to tell you what your risk of various diseases. What's the utility of really sequencing the entire genome?
You should, so the question is, if the HapMap and these reference databases can tell you about almost all of your variation, what's the use and value of getting your genome sequenced in a personal setting? So I guess there's a couple of answers to that. First of all, we don't quite yet know what the value of getting your genome sequenced in full in a personalized medical setting is, because for the most part, we don't know how to interpret the information that's contained in it. So there might be a lot of information. It is clear that while almost all the variation that you carry is common and can be assessed, it may be the case that for many medical phenotypes, and it's certainly the case for severe genetic diseases, that it's the small amount that's extremely rare or private in an individual that's more relevant to those medical outcomes. And one thing that we don't know yet, but we're aiming to discover, is ultimately how much of any particular phenotype is contained, has this information contained in the majority category, which is the common variation, and how much of it is in the rare and private component that might be just in your genome or in your family's genome. That answer will vary depending on what disease or phenotype you happen to be studying, of course. And ultimately, there's also the reality that we know most of the phenotypes and diseases we study from monozygotic twin concordance are far from 100% concordant. So that puts a hard limit on, even if we were super omniscient, how much predictive value there is in your genome. Probably we should stop and move on to MPG. So thanks.